Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing TIFR Biology question paper conducted in the year 2013. If you are new to my channel, please do like, share and subscribe. Coming to the year 2013. Coat color in cattle is determined genetically. The progeny of a cattle pair having a coat color consisting of red and white patches shows the following ratio with respect to the coat color: 25% white, 25% red, 50% red and white patches. Based on these data, which of the following statement is correct? Last year only we have discussed one question with almost the same answer. That is, they are codominant. So red and white may be equally well expressed or are codominant. Then next one. Male honeybees have larger eyes and larger antenna than the fertile female honeybees. Which conclusion on honeybee mating behavior can you draw from this observation? So male honeybees, they have larger eyes and larger antenna than fertile female honeybees. So which conclusion on honeybee mating behavior can you draw from this observation? Queens fly faster than drones. No. Queens have better vision than drones because their small eyes are sufficient for flight control. No. Drones use sex pheromone to attract queens. Drones must compete to detect the queen. That is more correct because they have larger eyes and larger antenna than the fertile female bee. So they must compete to detect the queen. Answer is option D. Then Empedocles hypothesized that we see object by rays emanating from the eyes. Which of the following will disapprove this? A blindfold will not allow you to see an object. You need a light to see an object at night. So they have asked which of the following will disapprove this hypothesis. So if rays were emanating from the eyes, then you need not have to use a light to see an object at night. So the answer will be option B. Human red blood cells cannot grow in culture because RBCs are terminally differentiated cells lacking nuclei. Answer is option C. A motor protein hydrolyzes one ADP-P bond to generate the physical force of 5 piconewtons and takes an 8 nanometer step at room temperature. What is the information required to estimate the efficiency of the motor? Energy released from ATP-ADP hydrolysis. So if that value is also provided, then you can estimate the efficiency of the motor. The next one. How many different transfer RNA molecules are possible in a hypothetical organism where the ribosome detect quadruplet instead of the triplet codon, including start and stop codon? So the usual 64 codons are formed by 4 raised to 3, that is 64. Since codons are triplet. Now they have asked if the codons are quadruplet, we have 4 nucleotides. There will be 4 raised to 4 possibilities, so it will be 256. Answer is option D. Which of the following does not contribute to protein diversity? Variants of the same protein. RNA editing. Yes, it will contribute. RNA splicing. Yes. RNA interference. RNA interference does not contribute to protein diversity. It is for inhibition of some mRNAs that are produced. So it will result in not protein diversity, but the protein produced from one particular mRNA will not be present or will be reduced as a result of RNA interference. Then, a competitive inhibitor of an enzyme, it increases the Km whereas the Vmax remains the same. So answer is option D. A scientist studying courtship behavior hypothesizes that while courting female beetles, male displays their horn as a signal of their health. If he is correct, which of the following must be true? Females can estimate male horn length. Yes. That is correct. Female appendages are not affected by their health. Females always mate with larger males, not larger males, males with longer horns will be chosen by the females. So answer will be A. Females can estimate male horn length. Now next one. A scientist spread bacteria on a nutrient agar plate and after some time transferred the resulting bacterial colonies to 3 other plates such that the relative position of the bacterial colony was same in all 4 plates. The fresh plates all contained the same antibiotic. The scientist notes that on each of the antibiotic containing plate, a few colonies survive the antibiotic. Which of the following is most likely to be observed? So what is this replica plating? Now this is your master plate. This is the sterile velvet. You just press that on top of this master plate and you transfer the colonies as such to another medium or to a fresh medium. And these are the colonies that you see in the master plate and these are the colonies that you see in the second plate where you transferred from the master plate. Here if you see the number of colonies is the same and the position of these colonies is also same in this replica plate. So the answer here will be both the number and position of resistant colonies are same on all the 3 plates. It is replica plating experiment. So the answer is option C. Then next one. From 1995 to 2007, Mycoplasma gallisepticum infection spread through the house finch population in the US. A student sequenced a 128 kb stretch of DNA from Mycoplasma strains in birds collected before, during and after the epidemic. With these sequences, he estimated genetic diversity in Mycoplasma at different time points, comparing it to diversity in strains from other infected birds, chicken and turkey. The graph below shows his data. What could have caused the decreasing genetic diversity in Mycoplasma strains from house finches if house finch mortality increased as infection spread? Now this is the genetic diversity. This is chicken, turkey, house finch and house finch Mycoplasma strain. They have enlarged the graph in this region, that is this house finch Mycoplasma strain. And what did you observe? The graph below shows his data. What could have caused the decreasing genetic diversity of Mycoplasma strain from house finches if so? What is the reason behind the decrease in genetic diversity of Mycoplasma strains from house finches? So selection favoring a more virulent strain of Mycoplasma. That could be the reason. Recombination between Mycoplasma strain from turkeys and finches. Now recombination, it favors genetic diversity. So option B cannot be the answer because it is said in question that genetic diversity decreases. So the answer is naturally option A. Then next one. In zebrafish exhibit horizontal stripes of pigment cells in the skin. A small fraction of a zebrafish population grown in a pond that is contaminated with a mutagen exhibits blue spots in addition to the horizontal strips. DNA sequence analysis confirmed that cells within the blue spot possesses a mutation in a gene that controls pigmentation. However, when these blue spotted fish are crossed to the normal wild type fish, these spots do not appear either in the F1 or F2 generation. That means these spots do not appear in the progeny. So it is not any germline mutation. This can be explained by the fact that mutation leading to blue coloration is gets correlated in the subsequent generation, no, gets corrected in the no, it is not appearing in F1 and F2 generation. It's a recessive mutation, no, it's a somatic mutation. That would be more correct. That is why it didn't appear in the F1 and F2 generations. In Mendel's experiment, the spherical seed character is completely dominant over dented seed character. If the characters for height were incompletely dominant, such that capital T are tall, capital T small t are intermediate, and small t are short, what would be the phenotypes resulting from crossing of spherical seeded short capital S small t plant to a dented seeded tall small s capital T plant? So capital S small t will produce the gamete capital S small t, and this one will produce the gamete small s capital T. Upon crossing you will get capital S capital T small t. So what would be the phenotype? It will be spherical seeded with intermediate height. So all the progeny would be spherical seeded and intermediate height.